we're going to get right back into this lecture, and we're going to talk about capsules and slime layers. We're then going to talk about flagella. We'll talk about Fimbriae, Pili, what else? Some other internal structures like ribosomes. And we'll finish off, we'll stop once we get to the eukaryotic cells, and then we'll do our last lecture on eukaryotic cells, okay? So the glycocalyx is basically we think of it as a sugar coat. It's a structure that surrounds the cell of the bacteria. It surrounds the cell wall, it surrounds the plasma membrane, so it just covers the entire cell. And it has two major structures. It can form either a capsule or a slime layer. These can help cells to be protected from immune systems, from external, potential damaging agents. They can also help cells to adhere to surfaces and then grow and form structures known as biofilms. So on the next slide, I'll show you what a biofilm looks like. So biofilms are structures that are formed continuously. They're formed in nature. If you've ever went to a pond and you touched a kind of slimy feeling rock, that slimy feeling was a biofilm forming. If you've ever had to clean out your sink or your tub and you feel that gooey stuff that's in there, that's a biofilm that forms. Every single day you have to clean off biofilm from your teeth. So plaque is a biofilm, and every day you scrub that plaque away. So biofilms form as bacteria grow and produce chemicals. Then other cells come in and they attach, and eventually you have this community of cells living together, growing together, that are protecting each other. And once you have a biofilm, it's very hard to remove it or to get rid of it, except for with mechanical removal, which is why when you have plaque on your teeth every night when you go to bed, you've brushed your teeth feel nice and smooth and clean. Then in the morning, they have that kind of gritty feeling. That's a biofilm already starting to form, so you brush your teeth again. Biofilms can form in wounds. So if you've ever had a wound that had that green pussy looking stuff on it, and so I don't know if this has ever happened to you, but it happened to me before. Your mom has to clean that out. They take a warm washcloth and they kind of scrape that wound to get all of that pussy stuff out. They have to do that because otherwise the bacteria will grow in there and cause a worse infection. They can also form, and so, I mean, this is not so bad because this is a surface wound. Clean it out, you keep it clean, and it'll go, the pathogen is gone, the wound will heal. If you have a deep wound, a deep tissue wound, and that biofilm is there, then it can actually allow for the bacteria to potentially get into the bloodstream. So that's really dangerous. Biofilms can also form on medical equipment. So you can have biofilms form on implantations. So say you're putting a hip in, you're replacing a hip, or you're putting a pacemaker in, or even something as simple as a catheter could potentially have a biofilm on it, and that can cause a lot of damage to the patient. So biofilms are very important medically, but they are naturally occurring in the environment, and their job, bacteria don't go out about their way to try and kill people. That's not their job. They don't care. They just want to survive themselves, right? What the biofilm does is it protects the bacteria. So in the environment, they need to protect themselves, and so to do so, they form these biofilms. Some bacteria, Didn't want that yet. There we go. Some bacteria have flagella as well. Flagella are structures that form from the cytoskeletal filaments, and they function in movement of materials. So here is a single polar flagellum, and this cell here has flagella all around them. This is known as peritrichous flagella. This is a single polar flagellum. Here we have Helicobacter, which has multiple flagella on one side. That's called Lophotrichus, I believe. I have to look up the name for that again. Lophotrichus might be both sides. But what they do is the flagella actually allows them or helps them to be able to burrow through the stomach lining, the tissue, the epithelium, to get to the lower layers and cause gastric ulcers. So bacteria utilize their flagella to help them to move either towards a chemoattractant or away from a repellent. So typically bacteria are moving in what we call runs and tumbles. And so cells are going to move in one direction very quickly, and then they kind of tumble around. They don't just move in one direction nice and easily. They do this tumble, run to get to wherever they want to get. There are other mechanisms that can allow bacteria to move or can stimulate bacteria to move. Aerotaxis, so oxygen, magnetotaxis, thermotaxis, or phototaxis. So these can all stimulate bacteria to move either towards or away from the stimulus. Pili and Fimbriae. So Fimbriae are these structures that are found throughout or surrounding the bacterial cell. Fimbriae are typically considered smaller than your pili, and they're both smaller than your flagella. If the cell has certain types of genes, they can form a very long Fimbriae known as a sex pilus. So that's what this is. This is a sex pilus. The sex pilus is a structure that allows two cells to attach to each other so that they can share genetic information. And Fimbriae function in attachment to surfaces. So the sex pilus functions in attaching to other cells, whereas Fimbriae just allow them to attach to surface, and then they can move along the surface. Internally, bacteria have a single chromosome. Almost every single bacterial cell is singly or contains one single circular double-stranded DNA molecule. Now, I say this, and so you may think, wow, that's not a lot of DNA. But this one cell here, this is all the DNA that came out of it. So, I mean, there's a lot of DNA within one single cell, and it's packed very tightly in a region known as the nucleoid, which is just a region within the cytoplasm. It's not, things can still move into it and out of it, but it's just where all the DNA is found. Sometimes bacteria have extra chromosomal DNA known as plasmids. These are also circular DNA, but they're tiny little structures that have only a few only a few genes. But these genes can actually allow bacteria to be resistant to different substances, or they may give bacteria certain characteristics that they've never had that allow them to be more likely to survive in different environments. So plasmids are beneficial, but they are not part of the chromosomal DNA. All bacteria have ribosomes. Ribosomes are the site of protein synthesis. Bacterial ribosomes are smaller than eukaryotic ribosomes. So if you look at, here's a eukaryotic, Wow, I can't talk. Eukaryotic ribosomes are called ADS ribosomes. The S stands for Sandburg units, and that's just the individual who found the sizes, okay? Each ribosome has two different subunits. You have a large and a small subunit. The large subunit of ADS ribosomes is a 60S subunit, and then we have a 40S subunit, okay? For bacteria, they are called 70S ribosomes, and they're made of 50S subunits and 30S subunits. So you have the 50 and the 30 come together, and they form this 70S ribosome. So please note, I'm not adding the 50 and the 30 together to get this number. I'm not adding anything. I'm just saying this is the top, this number here is the bottom, but when you put them together, they make up a 70S ribosome. So Sandburg units is the number that if you were to use a centrifuge and spin them out, that's when they would settle, is at that 70 Sandburg units, okay? And so it's on here, so I want to say it. We oftentimes target ribosomes when we're trying to kill pathogens, because if we can target the ribosomes and they can't produce new proteins, then they can't replicate, and then they will die. Or our immune system can take care of them. Bacteria also have a cytoskeleton. It's different from eukaryotic cytoskeleton, but it has similar functions, okay? Still is helps in maintaining cell shape and likely involved in cell division. There are storage granules. So we have these little tiny structures that hold nutrients and may even hold waste products to get so that they can be released. But mainly probably hold a lot of nutrients. There are gas vesicles. Gas vesicles allow cells that are aquatic to be buoyant so they can move. So if you have more gas in you, then you can move up and then you can get nutrients, or you can release gas to move away from a potential predator. The last thing that we'll talk about then are endospores. So endospores are a type of dormant cell that can persist in an environment for hundreds of years. They just stay there. It's like they're in a deep slumber. And when environmental conditions become favorable, the endospores can germinate. And so endospores can be found anywhere. They can be found in your laboratory. They can be found in dirt. They can be found all over. They're super resistant. They can resist boiling temperatures. They can resist dehydration. They can resist lots of different chemicals. And then they'll germinate only if the environment is favorable. So these guys are often thought of as a potential for biowarfare, which is scary. All right, so let's look at this cell over here. This cell has entered an environment that is unfavorable to it. So it replicates its DNA just like normal via binary fission, but it asymmetrically divides, forming a smaller structure and a larger structure. The smaller region is known as the forespore. So the cell, then the cell wall forms, but we end up having this forespore that has a membrane around it, and then we have that inner membrane. Between these two membranes, peptidoglycan is laid, a nice thick layer of peptidoglycan. So multiple layers of peptidoglycan are formed around it. Then some chemicals take away water from here and add calcium. This basically dehydrates the spore, Then the spore is released when the mother cell degrades. And this now endospore can survive in the environment until favorable conditions occur. So we're going to stop here. It's been a 15-minute video, and we'll get into our last video of the eukaryotic cell next. Bye.